But today is absolutely gorgeous fall day, and we see the weather at the weather moment. And but this is an absolutely gorgeous fall day. The colors are popping, the yellows and the reds. The leaves have not yet gone. We're about to get the bare trees shortly. But make sure you get out this weekend to get your hikes in before the winter comes in. It is, as usual, a pleasure to welcome you all here and those online also to another one of our fantastic speakers series members. Before I start, I will do my usual explanation of who we are, the Institute for Integrative and Innovative Research. I'm the founding executive director for that, Ranwu Jung. And you all know this, that we are driven by a mission to solve wicked problems, those that can't be solved easily, by a convergence approach, bringing together, and I think I'm going to start asking the audience to tell me how do we bring them together, right? The academic, industry, nonprofit, government, all of us pull together to find solutions. Really had a great meeting earlier this week on the rural health transformation part with multiple members from the state. They are all thinking about what can we do about that. So that's what we do. That's what we want. We want societal impact. We are living with the mantra of choosing grand challenges. Our grand challenge is integral health. We are looking to see how can we find solutions from medtech and food tech and cyber tech to find the solutions so that we can deploy solutions to get societal impact. So with all that said, do you guys remember that we had somebody called Purnima Madhivanan, a professor that came in to talk about things in public health? Well, today we have somebody who actually works with Dr. Madhivanan, and it is the example of how in our institute, when we have got medical sociologists and engineers working together, Jessica represents that. She has had the ability to take core, fundamental device development stuff and actually take that into the public health arena. And so you will hear from her later about how she has been able to bridge that, her students, her own research, making these partnerships that engage community, that engage researchers, that engage actual new device development stuff and entrepreneurial work. So Jessica Ramella-Roman, our today's speaker, embodies that. Jessica is I think, I don't know, there's a word for it that says somebody who can bring all these things put together. I can't get that word right now, but she's actually she got an electrical engineering degree from University of Pavia, Italy, and she still has her Italian base 100% with her, She earned both her master's and Ph.D. degrees from Oregon Health Science University from Portland, Oregon. And then she did a postdoc at the Applied Physics Lab at Johns Hopkins. Yesterday night, she was talking a little bit with another Johns Hopkins APL member, Ramella-Roman and Alejandro Gomez. And then she started at Catholic University. I was very fortunate when I was at Florida International University to have actually recruited her as a faculty member there. And so I've seen her grow the whole effort, but she has built at FIU and of course, the impact of it. She is a fellow of optics is what her basic area is. She's a fellow of SPIE and Optica and the American Institute of Medical and Biological Engineering, and basically a biophotonics researcher who is driven by needs of the community and developing the solution. So welcome, Jessica. It's a pleasure to have you here.
Thank you very much. Thank you. So first of all, thank you for having me. It's really exciting to see. I've never been to Arkansas, so it's a beautiful place. I'm really enjoying my visit, and it's always nice to see Ranwu and Jimmy are really good friends and a great mentor for me. So I'm excited to be here. So today we will go through many different topics. I hope that I will illustrate a little bit what I'm doing with my research. But first of all, I wanted to acknowledge my team. These are my two postdocs and graduate students. There are others that didn't fit in the picture. They are represented by the minions. And I have many undergrads and master students that are working hard on this topic of determining the risk of preterm labor. But before going there, I also want to say that I have basically two lines of research. And one substantial one is the development of wearable devices to manage cardiovascular disease. I'm part of a ERC called PATSUP, where we're trying to develop new technology to manage the risk of cardiovascular disease in underserved population. And in this context, I do a lot of modeling, I do development of optical phantoms, and then some instrument development as well. But let me go back. Today I will talk about my research on women health, and particularly, so we have many approaches towards women health. We have basic studies, and then we try to translate our knowledge to the clinic and even at the point of care. So let me tell you first a little bit about our problem. We are looking at spontaneous preterm birth, which is the delivery of a child before 37 weeks of gestation. Is the highest cause of infant death worldwide, and in the US particularly, it's a huge problem. In between 8 to 15% of all children are born premature in the US, and 50 million child worldwide are born premature as well. The wicked problem is that these instances are not decreasing; they're actually increasing. So worldwide, this is becoming more and more of an issue. And it's a very complex issue because the etiology of preterm birth is very complex. So it's due to infection, inflammation, there is poor nutrition, there are genetic predispositions to it. So very hard to tackle. Let's just have a problem with it. So let's see how it's diagnosed right now. There are ways that are, again, not very effective. The typical way is to look at the cervical length. If the cervix is shorter than 2.2 centimeter, then the woman is considered at risk. But even there, we were reading articles recently, and we find out that different races have different cervical lengths. So it becomes an issue of, you have to be aware of who you're monitoring, right? There was an assay, fibronectin, to look at cervical secretion. It was FDA approved, and then the FDA revoked the approval because it shows that they were noticing that it was not, the efficacy of it was not enough. And then, of course, there is the old way of checking the cervix stiffness through your finger. And this is really a way for us to look at what we call cervical remodeling. So at the mechanical composition of the cervix, your finger is very sensitive to it, and physicians are very aware of what to look for. But again, it's a very primitive, let me say, way to look at a problem this complex. So a little reminder of where the cervix is. The cervix is at the end of the uterus, and it's a structure that helps to keep the baby in uterus, right? So what we are looking for, the problem that we are tackling, is really a problem of cervical insufficiency of a weak cervix. So when the cervix is so weak that the baby comes out from the uterus prematurely. What is the cervix? Is this structure that has these two functions, right? One is to keep the baby in uterus, but the second function is to become compliant very quickly so that the baby can come to the world. It's composed of a thin cellularized epithelium, very, very thin, so that we can actually see through it. And then a cervical stroma, which is made of fibrillar collagen at the 70%. So it's very, very collagen rich. 2/3 are type 1 and 1/3 are type 2. And then there are protoglycans, there is elastin, there are smooth muscle cells. The problem that we are looking at is this remodeling, right? So the changes from the rigid structure to the compliant structure. The little graph here on the bottom is the remodeling process in a mouse. So the cervix is very stiff at the beginning, and then starts softening, and then becomes super soft, and the pups come to the world. There are stages of this process that are called softening, ripening, dilation, and repair. And if you look at the repair process at the end of gestation, you can see that it's within 24 hours. So this is an awesome thing. If we can do that to the collagen in our face, this is a $1 billion problem. And, this is exactly what we're looking at, this transformation, right? This remodeling process. Now, what we are trying to show is that the remodeling can occur prematurely. So, and the softening occurs at a time too early for the pup to survive, or for the children to survive. So we are really looking at the changes in this mechanic at an early stage. Where should I point this? Over there, right. Here we go. I was probably. So what we are leveraging in our modality is the fact that there is this richness of collagen, right? And as you know, collagen is fibrillar. In collagen type 1 can be a fibrillar structure, which has a hierarchical anisotropic structure, right? So you go from the triple helix structure at the molecular level all the way to fibers, to fibrils and fibers that basically augment the anisotropy of the environment, right? In the cervix, this is the cervix as you see it from the vaginal canal. The cervix has a circumferential alignment of collagen all around the aperture, right, which we call the os. And then there is a longitudinal structure instead on the side and at the os location. So really what we will leverage is that circumferential structure all around the os, which has been shown over the years by many different researchers through MRI with two-photon microscopy, second harmonic microscopy, and so forth, and microscopy in general. So what we are looking at is this arrangement, but also atypical changes of the extracellular matrix from this very arranged structure to a very disorganized structure. And you will see this map a lot, so I figured I will introduce it here. This is a map of orientation, right? Each one of those colors represents an angle. So red is 180 degrees, and cyan is 90, right? So we will follow this color map in terms of angular orientation of the fibrillar structure of the cervix in these locations. So the problem that we are trying to tackle are how does this remodeling occur? We know very, very little of the remodeling of the cervix in women. So we are doing a lot of, at this point, a lot of animal studies just to really understand the changes day by day of the cellular, of the ECM, right? So how does that connect to mechanics? There are many researchers that are really focusing on the mechanics of the cervix. And then finally, in my case, I'm trying to understand, can we leverage this information to predict preterm birth? So this is our approach. We go from very, very high resolution microscopy, the highest available right now, which is multi-photon microscopy. We use a particular kind of microscopy called the Murine microscopy. It's a polarization-sensitive microscopy. But we have also added this modality, the Mueller information, to colposcopes, so we can do it in the clinic with physicians. And then finally, what we are trying to do is bring all this to the point of care and allow really women to almost measure themselves, right? And or some lay personnel to do this at the point of care. And what I want to really point out that we think that this information, this fibrillar structure assessment through polarization technique, although of course at the microscopy level, it's fantastic, we can see the single fibril, this information is maintained all throughout the resolution levels, right? So even when we image the entire cervix at 3 centimeter field of view, we can still see the same type of information. So I think everybody by now knows a little bit about multiphoton microscopy, but this is the first technique that we use for this process. And the beauty of multiphoton microscopy, really, if I can just shorten it, is that it allows for a gated imaging. So you will only image the information at the focal at the focal point, right? So you, unlike confocal, where you can see on the left hand side, you see the light, spreading, scattering in all the direction as it enters the medium and goes to focus. For multiphoton microscopy, only the photon that combines at that focal spot are seen, right? So due to the two photon effect. Similarly, the similar effect is also for second harmonic generation. So we only see the information at the plane that we are interested in, which allows for much clearer imaging, of course. The drawback, of course, is that you need this femtosecond laser, which is very expensive, and then it's also a type of modality that it's hard to do in vivo. But anyhow, second harmonic microscopy is now pretty much the gold standard if you want to look at collagen. And again, it leveraged the fact that can only see structure that are anisotropic. And so it's perfect for collagen, for fibrillar collagen, because it has its own anisotropy, and you get this coherent summation of the signal from the molecular all the way to the fibrillar structure. So you can really visualize collagen in a really wonderful way. Now, let me just say that through a collaboration with a group in France, we have started looking at another form of second harmonic microscopy, which is polarized second harmonic microscopy. So in polarized second harmonic, what you do is that you impose a polarization state at the input light, a linear state. So it's actually not very difficult to do. You impose it, and then what you get is the modulation of the second harmonic intensity as a function of azimuth due to the azimuth of the material, right? So you get this beautiful lobing structure, which you can, through physics-based model, you can then use to characterize the azimuth or the orientation of collagen now at a resolution which is unmatched at 500 nanometers for collagen. So we have used this to look at the murine cervix, so the structure of the cervix in a mouse model. We look at samples at different days of gestation. In this case, what we have to do is cut the cervix at different depth. And then we have this setup. Again, this is pretty typical for second harmonic. You have a Tesafire laser, but the difference here is that we impose polarization right before the objective, and then we collect the information, both second harmonic and two photon. We can do that in a reflection, but in a transmission, but also in reflection. So this is the current best pixel size, so 420 nanometers. It's super useful when you are trying to image the collagen orientation at the of the cervix, right? So there is kind of a process, of course. Given the fact that our resolution is 420 nanometers, what we do is we get small patches, and then we have to tile this entire information. So some of these images I started doing when I was on sabbatical in France, and it was beautiful because you are back to be a graduate student, and so I was taking these images. Sometimes it would take me half a day, just to take these images. So it's a powerful technique, but it's obviously it's not optimized for human. So let me just tell you what we did, though. We look at the mouse pregnancy, and the reason why we look at mouse pregnancy is because, of course, mouse models are very common. But in terms of cervical imaging and cervical assessment, they are considered very similar to the human because the components, the collagen component is very similar. It's also the remodeling is very similar. Of course, there is a pressurized fluid filled cavity, so it's some of the mechanics are similar. And of course, the beauty is the very short gestation period, so we can do everything in 18 days instead of 9 months. And also we can do other things to the mouse, right? So what we do is we dissect the mice, we fresh freeze the tissue, we cryostat the sections, and then we create our slide, and we look at, again, different days of gestation. This is how, and I'm not sure it's showing very much, but this is how the polarizer, can we maybe switch this off, the lights? This is how the collagen looks at different input state of polarization. And then this is how a cervix of the mouse will look in 2-photon and second harmonic. So 2-photon is red, and which looks primarily at elastin and cellular structure. And then the green is second harmonic. So already just in second harmonic imaging, you can notice how there is this arrangement of collagen all around the aperture, the os, right, of the mouse cervix. You can leave it off because it's the imaging always requires a little more contrast. So we can see this beautiful orientation even with these images. But yeah, maybe you can look at that screen over there. Yeah, when you go to second harmonic, to polarize second harmonic, really this pops out so beautifully, right? Again, remember the azimuth map. We can now really recognize the angle as a functional position, and this is again at 400 nanometers resolution, right? So we can see that fibril really, really well. And again, if we look at the fibers, we start seeing this beautiful arrangement. This is early days of pregnancy, so it's the cervix. The cervix is really well arranged, right? The architecture is very clear. But if we go to later days of gestation now, we can see something different, right? Again, so the top are polarized and second harmonic, 2-photon and second harmonic imaging, and the bottom are these orientation maps. Again, probably look over there. And you can see how, at day 6, the collagen is really well arranged, but at day 18, in this case, collagen has lost its orientation completely. It's still there. We still have collagen in the structure, but it's randomly oriented now. Now, how do we quantify that? Is we have a imaging process where we calculate the statistics in a small region of interest throughout the cervix. So we use circular statistics for this. We look at the parameter like entropy or circular variance, which tells us how oriented, right, the collagen is. So as a reminder, for example, for circular variance, if you have a variance of 0, it means the collagen is completely arranged, right? That is one angle. And if you have a variance of 1, it's the opposite. It's completely disorganized. So when we do that and we calculate this variance map, now you can really see, even in this screen, how collagen is really, really arranged at early days of gestation around the os. And look at here, now it's not that it's not there, but the order is completely gone, right? So now we can actually quantify the changes in order, with this technique. And we've done it for many, many different days. And when we look at the statistics now, and in this case, we're looking at non-pregnant, day 6, 12, 15, 18, now we can see that what happens is that up to day 15, the cervix is really, really nicely arranged. And after that, we go in a different remodeling stage where the orientation has has been lost, right? So the mechanics also have been lost. There is a really strong softness at that point. So we published this not too long ago. So if you're interested in the detail, please look at this paper on the ACS Botanics. Now, I love this technique. One, because the images are absolutely beautiful, and I'm a sucker for that. So I love beautiful images. But of course, it's a second harmonic system like this, costs almost $1 million. So and it's almost impossible to do it in vivo, right? I mean, we're looking at strategies, but it's very, very hard. So as much as I like this technique, there are other techniques that we're trying to push that are more, human and animal friendly. And the one that we'll talk to you about now is this Muller matrix microscopy, where we can do human studies. So this is a microscope. We still, in this case, this is a bit special. We still use this very sophisticated, expensive laser, let's say, this Tesafire laser. But what we do is we combine the second harmonic information, the 2-photon second harmonic information, with this Muller technique. The idea really here was to show that Muller and second harmonic give very similar information. So there are a little bit of detail here, but we can do several things here. We can do reflectance Muller, transmission Muller. We can do confocal Muller, which is kind of cool. And we can also see the entire cervix in one shot instead of having to tile small pieces of that cervix. And again, this week we published this extensively. The one thing with let me go into this small math break here. To obtain Muller microscopy, basically you have to measure the transfer function of polarized light through the material, right? So what you do is that you modulate the light source at different polarization state, and then you collect the same polarization state at the detector site. So it's a combination of 16 measurements. So 4 input states, 4 detection states, 16 measurements that give you this Muller information, this matrix, basically. And then from there, you can decompose the Muller matrix. And the decomposition of the Muller matrix allows you to measure things like depolarization, linear birefringence, which you're probably familiar with, and the attenuation, and particularly allows you to measure azimuth, right? So this angular information. So what we get, again, is images like this, where we get the second harmonic information, but we also get the linear retardation, the azimuth, all that in one shot now, and they are co-registered. So what we can do is many, many different things. One cool thing that we did was to show that we could visualize elastin just based on this Muller information. We published that work earlier on. And then we can do depth resolved, we can do confocal, right? And we can start looking at this gestation business with this simpler technique, right? And getting information that are actually quite similar, right? In terms of the angular information, you can see here, and the collagen, you will recognize this arrangement around the os. So when we do this work just based on Muller, we get very similar information. I hope you remember that we were measuring different gestation day, and here again we can see that they at the day 15 and 18 things, the circular variance information is completely different from early stage of gestation, right? You get this big hit of the remodeling. So Muller and polarized second harmonic can give us similar information, this is really hot off the press. This is a very cool thing that a student from France that is visiting my lab is putting together. We can also now train this system through the ground truth, which is second harmonic, we can train the system to recognize collagen, uniquely collagen, in the Mueller information. And it's, which basically allows us to train any type of microscope, not just this special, combined microscope. Any type of microscope can now visualize collagen uniquely through machine learning. I thought that this was super cool. I just wanted to add it. So we had the super cool microscope, and then we have the poor man microscope, which is the much simpler technique. In this case here, we have a white light source that we can filter at any wavelength. And again, we do Mueller in a sense that we can impose state of polarization at the source, and at the detector, we can do reflectance or transmittance. But the main thing is that this system does not have second harmonic, right? It's just a microscope. You can do this in your lab. You can take a microscope and impose polarization state and obtain Mueller information. So it's now much more accessible, let's put it this way. Again, for Mueller, you have to do this decomposition, you have to get the decomposition information, but at the end, we really get the same information. Again, these are the maps of collagen remodeling of the cervix. And if we compare this to the polarized second harmonic, we can see that it's a little bit different, right? Because again, second harmonic only shows collagen. Mueller shows everything, but it's very sensitive to collagen. So from here to here, for you, probably it's, you're not convinced, right? There's something here, but it's hard to see. But if you actually isolate that information through, machine learning or through knowing where you are looking at, now you really get the same data, the same powerful azimuthal assessment, right? So we now can do the same studies, but we have refined this human study more and more. So day 6 all the way to day 15, and again, similar result, even with this very simple, very $5,000 kind of microscope. So and the other thing this was the information as a function of gestation. So if we again look at the early stage, day 6 to day 14, compared to late stage, day 15 to 18, we see this big change in arrangement, in circular variance. We can also do that as a function of depth. So, this is the cervix of a woman, and this is what we call the endocervix, inside the area of the cervix, inside the closest to the uterus, right? And this is the ectocervix, which is the only part that we can really see clinically, right? We are not going to go all the way to the uterus. We can only see this end. So there is a real interest in trying to understand, is the information that we get here clinically relevant, right? Because if everything happens at the endo, then there is no point to do our technique. But we did this study, we looked at different depths of the remodeling. So again, they are collagen arrangement as a function of depth, and so we can see that again things change from day 16, 12, and 18. So there is this remodeling. But what we are noticing is that at the endo to ecto does not change drastically, right? The type of information that we get is quite similar. Maybe at this end here, when we look at day 12 and 14, there is a little bit of change. But if anything, we're more sensitive at the ecto, right? There's more remodeling at the ecto compared to the endo's part of the cervix. So we are, very reassured. Of course, this has to be duplicated in humans, but we are pretty convinced that we will be able to see relevant information on the side that we are sensitive to. And this is, another metrics, which entropy, and again, showing similar type of information. So finally, to conclude our, from the nano to the macro level type of information, we built these Muller microscopes. These are some of the graduate students that worked on it. Mel did the point-of-care device, and Joseph did the clinical device. So we went to the clinic and did a series of measurements on women that were at risk of preterm labor. And again, just to show that we think that we get very, very similar azimuthal information, even at this level, at this macro level. So this study here was looking again on, well, here even non-pregnant compared to later stage of pregnancy. And we had another metric back then called kurtosis, which is basically the same. It's the arrangement of collagen. And we could see that there was a difference, right? A statistically significant difference in between early or non-pregnant and later stage of pregnancy. And again, we could see this map of orientation in the human cervix and see how the statistic of that orientation really changed, right? Max peaked orientation for non-pregnant, and then this distribution as you go to later stage of pregnancy. And then from there, we were told we actually had quite a difficult time recruiting because nobody wanted to do a speculum exam. I don't want to do a speculum exam. I think if you ask any woman in this audience, they will say it's not pleasant. So why not try to do it without the speculum, right? The speculum is that device that opens the vaginal canal, right? It's a big, very often it's made of plastic, and you insert it through the vagina in the vaginal canal, and then you crank it, right? Crack, crack, crack. You open the vaginal walls. So why try to remove that and use a device that is more similar to a tampon, like in this case? So we built this imager. It's a one-click imaging system that has a silicone-coated inserter. This idea actually was, similar to, again, a tampon or a vaginal ultrasound, for example, that form factor. So we create this whole process to build this silicone sleeve. They are disposable. And of course, there is quite a bit of optical design here to try to get the same type of information and the polarized type of information. There are now these beautiful polarized camera that you can buy off the market that are actually relatively inexpensive. And they have 3 states of polarization at the chip level. So you can do a snapshot imaging of polarization. And so we, again, with a collaboration with a group I call Polyclinic, we develop models that allows us to get the same information that we get in the microscope much faster, right? By modulating the light sources here at 4 different stages, and then 3 different states from the snapshot polarized camera, we obtained the Muller information. We did a bunch of testing showing that the angle that we were getting was correct. So we have silicon phantom where we can do this. We patented this whole approach. And then here is, again, our angle distribution in a woman's cervix. Now we can see it really very well. So what we now doing is that we are doing pilot human studies with at UM and FIU, and we have started recruiting and look at the cervix of different women, both pregnant and non-pregnant. And finally, the last thing, the hot of the press is this one. This is a new approach of imaging, which is based, again, on a machine learning decomposition process, where we can get the azimuth information, the angular information, and the retardation and all that at 13 frames a second. And, I know you're not in a biz, but this has been, it's unseen. So far, the best was 2 frames a second, 3 frames a second. 13 frames a second puts us at a very comfortable position where we can do this, in real time, right? It's not 30, but 13 is quite acceptable. So we can do, it's now clinic ready, and we are we are recruiting, extensively, trying to bring the technique to the point of care again. So again, we are in the business of characterized collagen architecture. We think there is a strength in is a modality that has its own merit, that can see collagen at the site where we can see it through the vaginal canal, and the fact that, I mean, at the physics level, it's really interesting, the persistence of the information from nano to all the way to macro, right, to millimeter size. So we're doing a lot of fundamental studies on that. But that's pretty much it. I want to acknowledge, my group here. They are really, right now I have this fantastic group of students. I always had great students, but this is really awesome group. They did all this work. These are beautiful images that we're getting. Marie-Claire Shankline is the collaborator at Polytechnique. And this work is being funded primarily by Stroke, which is a STC center that looks at microscopy out of Boulder. And that's it. If you're interested, there is a lot more information on my website, and I'm here to answer any question you may have. So thank you. Or if you are in the optics, there is a conference coming up on women health in optics and polarized light in optics. And just come and visit us in Miami if you have time.
Fascinating talk, and obviously you all know we've been talking about what can we do in case of maternal health, and preterm labor is a major issue. And so, trying to see what we can do about bringing this technology here and seeing how could we potentially have it deployed both at the in clinics, but also if the campus level is available, then actually having it available in our rural health, rural communities where access to even the clinic is an issue. But open to questions and discussion. And you might perhaps also, as we are talking, Jessica, reflect on how you have also used some of this for cervical cancer detection. So, question open. Yes.
Hi, very exciting talk. So I had a few questions. So there should be a protein that is responsible for this reorganization of the collagen, right? Fiber. So when you're doing human studies, you check it against that protein to find the correlation.
No, we have not, but we should, right? So yeah, the human study has been so let me just say this. At some point, we were awarded an NIH grant to do exactly that. And then because of changing priorities, it was removed. So we had to remove that piece. But yeah, you're right. They will be very interesting, and something that we hope to do in the not too distant future.
So
through the fibronectin as well.
But do you look at it against ultrasound?
Yeah, we measure ultrasound. We do cervical depth, basically the thickness of the cervix. That's the other modality that we do. The one thing that we really wanted to do, and we are doing in animals, we're planning to do in animals, sorry, is to use shear wave ultrasound, which gives us mechanics. So the stiffness information right there. And but in the clinic right now, we're just doing ultrasound to measure the thickness of the cervix. But the thickness of the cervix is varies so much. I mean, it's hard to.
Fantastic way to scan. It's really important for the risk assessment. I have a couple of specific questions. One is regarding your mouse studies. So, which kind of mouse did you study for this experiment? Like black seeds or?
Oh my God, that's good. No, these are just normal mouse. They're not particular, not genetically modified or anything.
No, I mean, must be an inbred strain of mouse. So the reason why I'm asking is that we have several inbred strains of mouse, and there is a phenotypic difference.
Mm-hmm.
So I would like to know that if you had the time to see if this collagen fibronectin ratio and this repair remodeling process in the uterus or cervix changes across inbred strains, because that gives you a very good, strong phenotyping power. Because we see the similar events in when we see other fibrous tissues, as example, kidney or lungs. So,
yeah, no, I have not looked into it. Basically the mouse model was established by this other collaborator. She's actually the queen of mouse study related to cervical modeling, Ramella-Roman. She's in UT Southwestern. So we inherited the mouse model from her. I'm sorry, I completely forgot, but it's, you know, the run-of-the-mill mouse. It's nothing, nothing special. But yeah, it's a good point. I definitely need to have a look at that. The one reason why the next stage actually is to induce preterm on the mouse. There are inflammatory processes that we can induce and create a preterm situation in the mouse. So that's where we're going, and that's also one reason why we follow this very specific protocol, just because Mala has already done it, and we are going to, without imaging, but we were going to do the same.
And how do you foresee that? You know, there is a very established and robust method with cervical checks, and how do you foresee that this method is going to change how we forecast, you know, at-risk individuals, right? So, is there a clear change in the pattern of collagen deposition or fibronectin deposition that you can see or you can measure? How is it?
Yeah, I mean, if you recall from I mean, it's really based on, at this stage, it's really based on visualization. So let's go back to where we can really see it. To this, right? So we can, what we propose is that looking at the statistics of orientation in the cervix will help us the prediction. We already have a machine learning approach to it, where on the mouse we can actually tell you this is day 14 or this is day 12 pretty accurately on the mouse. But the old technique is based on this visualization, so it's really an imaging technique.
Yes, I noticed that you did not talk much about the Mueller imaging, because there are several arrangements of polarization between the emitter and detector. And does your system do everything in one shot, or you have to rotate the polarizer to get which polarization, cross polarization or something as needed?
Okay, so different systems do different things. In the microscope, we impose the polarization with this system here. There are liquid crystal retarders. So there are a couple of two liquid crystal retarder and a polarizer both at the source and the detector. And we change one at a time, but they're pretty quick. I mean, the switch of a liquid crystal retarder is 10 microseconds. So you can switch pretty quick. But of course, this is a scanning technique. So, you do a scan and switch, and then you go to the next, and you have to do it again, right? So at the microscope level, it's relatively slow still. But the very last slides that I showed you, so the snapshot imager, the camera itself has 3 states of polarization, 3 linear states. So that's one snapshot. The detector is one snapshot. And then the input state, they are LED. Well, actually, in this case, they're small laser diodes that is switched and very, very quickly. And so we do 4 input state, and then per each input state, we get one snapshot, which has already 3 states of polarization. So in that case, and then we can go with this approach, just with Mueller, we can do 25 or so frames a second. We can acquire very quickly. But then the decomposition, it's a very timely process, right? Because you have to do it pixel by pixel. But with this new approach, this machine learning approach, we can do the decomposition with machine learning and then get the 13 frames a second. I'm so excited about.
Did you have to apply the Chapman decomposition and the machine learning? Couldn't you apply the machine learning without the
No, it's without the decomposition.
Without the decomposition?
Right, exactly. So the machine learning gets away from the new Chapman decomposition, which is, again, it can be accelerated, but it's still time consuming. But the machine learning does not use at all and I'm not an expert in machine learning. I want to point this out right away. But it seems to get exactly the same information and in a much quicker way.
So that device that you showed, the small one, that's what when you call the Mueller is 3x4?
It's a 3x4, but this we have shown again. We have filled up the last row basically. This is a work with the other collaborator, Rans Vigor and Tatiana in France, and we can fill the last row. Also, in reality, and this is my sense, is that for azimuths, if you're only looking at azimuths, all you need is a input state which is circular, which is clearly circular. And then you can do it with the linear state of the camera. You don't really need even the mirror metrics. So it's for many tissue that will work. If you don't, basically the last row gives you the attenuation rate. It's and circular the attenuation. And if you don't need that, then
I have many questions, but I am saving to the meeting with you.
Oh, sounds good. Sounds good. Yes.
Yeah, thanks for the talk. Really interesting. I had two quick questions. One, looking at collagen structure, the orientation is certainly important mechanically, but also just the density of collagen and the cross-linking and the maturity. People sometimes try to use second harmonic to talk about like the maturity of the fibril bundling. Have you tried to look for any of those other collagen properties? Are you interested in those sorts of things as well?
Yeah. So actually, I couldn't really present it today because because it's work that was done primarily in France, but in the second harmonic, in the polarized second harmonic data, and we're trying to publish it into super journals. So, anyhow, in the second harmonic data, we are looking at gaps, and it's really clear how the gaps change at different gestation day. So and other people have looked at gaps as well. So, but with these modalities, it's really quite clear. So yeah, we have looked at that. In terms of, we have not done other work in terms of cross-linking or looking at that, because to some extent was done by others, by Mala and others in this domain. So we didn't want to replicate that work. And then now what we're really interested in is try to add the mechanics in the live model. So we are in the process of building another system based on aspiration that would give us local mechanics, again, in the mouse model. There is already a company that sells a similar system for women in Switzerland, and they can do aspiration at a small region of the cervix and then correlate that with gestation time. But it's something that we're definitely looking into.
That's very cool. A second quick follow-up question. How much information, predictive information, I realize you're not quite there yet, but thinking about ultimately trying to predict what's going to come for this mother and baby, right? How much do you think the information is in a snapshot versus a delta between two snapshots?
Yeah.
And that's hard to look at in the mouse because you're obviously sacrificing it certain time steps, so you don't have the temporal time course.
Right.
Are you thinking about that? Like I could imagine different mothers having different baseline levels, and then the deltas are what are really important for predicting some final preterm outcome.
No, I agree that I think the delta would be a very, very powerful way to look at it. One reason why we're developing all this microscopy is also to look at the live mouse. So we are looking at doing the information on the same mouse rather than sacrificing the mouse at the gestation time. It makes sense. I mean, there has been some work looking at this remodeling in women, so in larger group, not necessarily with polarization, but with the shear wave elastography. And it's not as, I don't know, my feel is that there is a consistency of remodeling, right? Now, of course, in the pathological cases, I don't know if it's, you know, the quickest change or they start at a different slope, right? And maybe that it's a combination of those effects. I don't know if I am explaining myself, but yeah, that's why, you know, going human is really, really important to see how things change. Thinking, but not doing quite yet.
Wonderful story, and looking forward to the end when you get further on. You were excited about the 13 frames per second. And I'm curious, so with the system as it is now, how long does it take to get the images that you need? You know, like why is 13 better than 2? And
well, because you know, in imaging, when you when you do imaging in a human, unfortunately, they tend to breathe, they tend to move, they want to talk to you, and imaging is complicated here because of this. It all causes artifacts in the imaging, right? So if you're looking at the fibro that is, you know, the resolution really is in the hundreds of microns, and they breathe, then you know, your imaging will be an average of that. So in imaging in general, you try to go as fast as possible. Yeah. Right. Just simple as that.
How long does it take, though, like with the 13 frames per second or with the 2 frames per second? How long are you imaging?
Well, so the imaging process is the first thing is you have to find the cervix, right? Which is actually surprisingly not that easy, in the sense that people don't recognize, right? So because there are vaginal walls that are collapsing and other things. So mostly that's the majority of the time you're trying to focus, but we can do that 30 frames a second and look at where we want to image. And then the 30 frames a second allows you to then, you find the cervix and then you click your, you know, your snapshot. And, you know, in 1 second, you're acquiring 30 frames. So you can average the 30 frames or pick the, you know, depending on light condition and so forth, you pick the one that has a better contrast, it's not moving, or, you know what I mean? That's why it's critical to go fast. It's all of it is based on reducing motion artifacts. Really, that's the main thing. Also, you know, of course, in imaging, always if you average, you get better information. So you want to get as many as you can within a short time frame. And finally, you know, nobody wants to stay there with something certain in the ground for a long period of time. So you're always trying to shorten the time of acquisition.
The other question is about like what's known about this remodeling with multiple pregnancies. So like if a woman has had 2 or 3 kids before, is this a different story or is it pretty much the same?
Yeah, no, it's those are all the biological variable are, you know, enormous. So in fact, I was mentioning this yesterday, the women we are focusing on are the ones that are at risk, and risk is determined by you had another delivery that was preterm. So that's the highest risk, right? You're carrying twins. That's a very, you know, high risk. You're old, that's risk, you know, and so forth. So there are some, they stratify the patient already based on this risk. And then it's interesting to look at that as well, because, for example, in the mouse, we have looked at mouse that had delivered more than once, right? So they had already delivered, and then, so for example, we were doing these studies, and then we get this mouse, and it's completely different. we're like, what the heck? I know how is this mouse behaving like that? And it turns out that it was an older mouse that we had, and they had already had many pups, and now his cervix was completely different, right? So his staging was different. So yeah, the biological variable are not in our favor, I have to say. They have to be very controlled. But all this information together, I think, helps you.
Also, couldn't it be in that case you look at it before, because it's an in-person?
Yeah, so that's where the delta.
Individual is personalized to that particular.
Yeah, that's how I was saying. That's why the delta is maybe an approach to look, from normative all the way, early stage all the way, following a person over time is what we want to do.
Other questions on this area? Want to tell a little bit about what you did with the cervical cancer screening?
Yeah.
And the trial in India?
Yeah. So we are looking at cervical cancer as well. This modality has been used for cervical cancer assessment by other groups, and shown that they could, so cervical cancer, the staging of early stage to late stage is very important, right? You want to get the information as soon as possible, of course. So it's scene 1 is where really everybody is trying to work on as the early changes in the epithelium. And so we had a study with a similar images to the last one, but it wasn't insertable. So we were still using a speculum, and we went to India, where Purnima Mariman has a center to look at cervical cancer in India, where they have not already spread with use the vaccine, the HPV vaccine extensively, so they have much higher cases. So in that, we looked at a women coming both in a clinic, and also we brought our imagers on a pedestal all the way to rural area in some really very dramatic conditions on a wooden table, and it's just the whole thing of working in rural area was quite interesting. But we looked at I forgot how many, but we looked at this tried to stage cervical cancer, and scene 1 is difficult. It's something that we are working on, but later stage we can definitely distinguish scene 2 from scene 3, and now we are trying to push all the way to to this scene 1 goal, or even precancerous lesions. So they even visualize in the cervix. There is a group in Duke that has built a similar imager. Actually, we learned a lot from them. And they don't have any polarization, nothing. Just they look at the cervix, and then they again use ML, or they send it to pathologists, and even just looking at the cervix, it's advantageous in those areas, right? Where maybe you don't have a physician at the location, but you can take a picture and then send it, at a hospital for assessment. So that's the idea. We did a preliminary study. We definitely want to go back and try to do more. But at this stage, we're really focusing on trying to go to earlier stages of prediction. Even in the US, because we have the path, but then there is still it's not a 100% success rate, and so it's still a problem even in the US. I was looking at a statistic not too long ago. People still die of cervical cancer in the US and all over the world.
Okay. One last question.
Yeah.
So I'm curious to know, first of all, thank you for the great presentation. It's amazing. So I'm curious to know the commercial viability side of it. So if we consider it's a personalized point of care device, if I can label that, then is it just applicable to preterm females who are for whom it's second pregnancy? Right now, if we try to look at the market size, then is it that group of people right now yet?
Yeah, so it's interesting. Well, we did are you familiar with iCorp? Yeah, so we did the students did an iCorp on this, and there are different markets. there are people there are groups that are just interested in visualizing the cervix, even without the polarization information. So, and then the preterm market, the answer was primarily to look at women that are risk. So in that category, because if the way they described it was that if a child is born premature, is $1 million baby. So to take care of that child is going to cost the hospital $1 million. So any way they can do to avoid that cost for a hospital is very well. It's interesting to them. So, but we were very much directed to focus on groups that were already at risk, and maybe this could be sent home, and they, to some kind of medicine, or it could be implemented for that particular group. I answer your question.
Hey, so thank you very, very much, and I presume no questions online, right? So thank you, everyone. Fascinating. We hope that we are able to get this. You're meeting some other people from the community, the clinic things. We hope that we can get this started here, and then we'll be back here and we will be presenting joint data.
That would be
fantastic.
Great. Thank you for your attention. Thank you.